I worked on DNA repair and DNA replication. I started working on the PARP inhibitor mechanism of actions when I was in IFPOME lab, and then I moved to the MD Anderson Cancer Center within a biotech company that is embedded in the cancer center. Yes, so I am really proud to say that I developed a novel PARP trapping assay to be able to quantify and measure in a much more accurate way the ability of PARP inhibitors to trap PARP on the DNA, on the chromatin, and this is in cell lines. Yes, so this is something that was done by my colleagues, and it's a proteomic approach that is based on chromatography, and it's a new chemical proteomics that is able to measure the ability of one PARP inhibitor to bind to different PARPs, because we know that the major anti-cancer effect is given by inhibiting PARP1, but PARP is composed of a family of 17 PARP members, and we wanted to profile the number of PARPs within the family that are bound to each PARP inhibitor. Absolutely. So there are other cytotoxicities, other general toxicities in the clinic that we observe specifically for example, niraparib or rucaparib. And we did a screening to check whether there were some off-target effects not within the PARP family. So whether these PARP inhibitors targeted other components in the body, other proteins that are present in the body. And what we saw strikingly is that niraparib in particular has a very high affinity for transporter of serotonin, dopamine, and other neurological signaling proteins. And we believe that this, although at the moment this is really a biochemical test, it's not unreasonable to think that this could be correlated to effects like insomnia that are specific for these PARP inhibitors and not for others. Yeah, so what we measured is basically the fact that in the clinic, we observe certain toxicities, and the preliminary toxicity is the hematological one. So we noticed that several PARP inhibitors are more toxic than olaparib. But in an in vitro assay, we couldn't see these differences. So we wanted to check in vivo whether there was a reason for this clinical observation. And what we did was we tried to test if the compounds, the different PARP inhibitors, had a different way of distributing inside the rat body, the body of the rat. And basically what we saw is that olaparib does not go into the bone marrow. So it's the accumulation in the bone marrow is very limited compared to the plasma distribution, whereas the other PARP inhibitors, in particular talazoparib, niraparib, and also rucaparib in some extent, they accumulate into the bone marrow. And we believe that this is the reason for the increased cytotoxicity specific for the hematological component. So I have to say that although I'm very proud to report this, I am actually just a spokesman, and this work was done by a team that is global, because basically we are based in Cambridge, UK, but part of the team is based in Boston, in Gatehouse Park, in Waltham actually, in the US. And most of the time we have virtual meetings, and we really feel that every component, every person from this multidisciplinary approach team brought in this virtual table the possible best, and this is why this is really a teamwork that is multidisciplinary and multi-global.